Today we will hear stem cell research from our guest speaker, Dr. John Tisdale. But before I introduce him, muchas gracias. Now I am going to tell you a little bit about Dr. Tisdale. Dr. John Tisdale is a senior investigator at the Molecular and Clinical Hematology of the National Heart, Lung, and Blood Institute and the National Institute of Diabetes and Digestive and Kidney Diseases at the National Institute of Health. Most recently, Dr. Tisdale discovered a blood stem cell transplant regimen that reverses sickle cell disease in adults. This study was published in December 10, 2009 in the issue of the New England Journal of Medicine. As a result, the American College of Physicians has just awarded him the Richard and Linda Rosenthal Award No. 1 from the Rosenthal Family Foundation. The award describes Dr. Tisdale as physician scientist whose recent innovation work is making a notable contribution to improve clinical care in the field of internal medicine. It is a real honor to have you here with us today, Dr. Tisdale. Welcome.
Thank you. Okay, so for the next 10 or 15 minutes, I'm just going to give an introduction to research on stem cells. I'll give you just a little taste of what we do at the NIH, but I wanted to keep this broad and try and stimulate some questions about stem cells. So many of you have probably seen in the popular media lots of references to how stem cells may cure a number of diseases that affect humans: diabetes, heart disease, Parkinson's, and I want to give you an update of where I think we are with respect to approaching these diseases with stem cells, and also give you a taste of some of the problems that have been encountered along the way. So first, for some definitions. There are several conventional sources of stem cells. The most widely used, the most widely researched are somatic stem cells. These are stem cells that you hear people talking about as adult stem cells. They're the seeds for growth for whatever organ or tissue that they come from. They're isolated from these tissues or organs in which they reside, and they're multipotent, meaning they can make all of the different types of cells within an organ or a tissue. But they're probably specific only to that tissue. So if you have a muscle stem cell, it probably only can make muscle and not some other tissue. If you have a bone marrow stem cell, it can probably only make blood, and so on. But there are those that think that these cells might in fact be pluripotent, meaning they can make all of the cells of the body. That's still very controversial. But suffice it to say, there are many established uses for stem cells, including bone marrow transplant. Bone marrow transplants have been performed now for more than 4 decades. This is a form of therapy where bone marrow stem cells, which reside in the bone, can be transplanted from one individual to another to cure a disease of the blood system, for example, leukemia. And here's a hierarchy of the bone marrow stem cell compartment. You can see stem cells here to the left have the ability to not only remake themselves, but to make all of the components of the blood. Lymphocytes, these are specialized cells for fighting infections. Granulocytes, these are other specialized cells for fighting infections. Platelets, these are the cells that clot the blood when we're injured, and red blood cells, those are the cells that carry oxygen. So transplanting these stem cells or seeds of the bone marrow could replace all of these blood cell components in a human being and be used to approach a disease of one of those components. Like I said, leukemia, our interest being sickle cell disease affecting red cells here. But if this pluripotency were true, this so-called plasticity, that bone marrow stem cells can actually make other organs, then this could be exploited to treat other diseases like diseases of the brain or the nervous system, type 1 diabetes, so on. This is just to show our recent work that was alluded to already. We developed a way of using bone marrow stem cells from somebody else who doesn't have sickle cell disease to partially replace that of a patient with sickle cell disease. This is a very severe form of anemia that, along with anemia, has multiple other complications that results in a very shortened lifespan. And here you can see we use a special method to get these cells in so they're not rejected. They come from somebody else. And over the 1 year post-transplantation, you can see there's normalization of their hemoglobin. This is 8, which is quite low, a level of anemia here, and this is the normal range of hemoglobin now after transplant. And this has also reversed all the other complications of the disease, most notably the recurrent pain crises that these patients experience. But as I said, there's been a lot of hype now that bone marrow stem cells might be able to treat other diseases outside of the bone marrow. So I show you a website here of one stem cell center that sprouted. Let's see if I can make this bigger. That sprouted to take this kind of technology onto. Is there a volume here? Let's see. I think it's probably turned down on the computer itself. Maybe so this is a stem cell center in Europe. Maybe you have the volume there. That's basically showing that bone marrow stem cells are going to be used to treat patients with stroke. So these are patients that have had a cerebrovascular accident. They'll go and get their bone marrow harvested and then squirted into the cerebrospinal fluid of the brain with the hopes that those cells will migrate and correct the damage caused by the stroke. I don't think there's any reason to expect that this will work. And unfortunately, there have been a lot of clinics that have sort of sprouted because of the interest around, I'm going to close it now, around this type of therapy. Let's see, it doesn't want to close now. Okay. And in fact, I had a friend call me after a stroke to see if I thought he should go to this center. And in fact, he did. Spent a lot of money, and of course, he didn't see any benefit. So there have been a lot of studies that have been initiated, I think, prematurely because of the hope that these types of stem cells might, in fact, be therapeutic. The other source of stem cells is a more controversial source, embryonic stem cells. Now, these cells are derived from the inner cell mass of a blastocyst. So this is a fertilized egg that starts to undergo division. And some cells can be scraped off here, put into a flask, and these cells can be grown into virtually any tissue of the body. Blood cells, heart muscle cells, neuronal cells. And of course, if these cells could be directed safely towards those fates, they could be used in regenerative medicine. So there's been a lot of interest over the last 20 years in developing this kind of therapy for humans. But these human studies have been encumbered by both technical and ethical issues that have really held up progress in this regard. Now, during the Bush administration, President Bush signed an executive order that, in fact, allowed research using U.S. federal funds to direct research on embryonic stem cells, so long as these embryonic stem cells the derivation had already been initiated in the embryo from which the stem cell line was derived was no longer intended to be developed as a human. So these were cells that were gotten from in vitro fertilization clinics, and that at least allowed stem cell research to proceed. But the biggest problem was that the number of eligible embryonic stem cell lines was severely limited once you applied all of these criteria to those that were in existence. There were about 60-something that were known, but there were really only about 4 or 5 that could be used. So investigators went on to try and figure out other ways to make embryonic stem cells that would comply with these regulations. So it turns out you can take a nucleus out of any cell, so you could scrape a skin cell off of me, take its nucleus out, take the nucleus out of an egg, put that nucleus in, and eventually derive embryonic stem cells that had my DNA in culture. That was first used to clone animals, and in fact has been successful in deriving embryonic stem cells from humans. But many objected because these could be in fact used to clone humans. So it was then developed that you could make these cells, but you could put a gene in that would prevent them from ever implanting. So you can make an embryonic stem cell line. Sure, it could be used for cloning, but it couldn't become a successful pregnancy if you stick this gene in. There was further pushback on that technique, so then it turns out you can make embryonic stem cells from an egg that you can start dividing without even fertilization. So that doesn't have the potential to become human. Or, in fact, you can take a cell, an embryonic stem cell line that exists, and fuse it with a skin cell or some other cell and make embryonic stem cell lines. So these were all new methods that were developed over the subsequent 5 years. But then in a push to really make adult stem cells more supported in this arena, President Bush then signed another executive order really trying to get federal funds funneled more towards adult stem cell research. So you can see here stem cells that are capable of producing all or almost all of the cell types of the body and may result in improved understanding and treatments of disease, but are derived without creating an embryo, destroying, discarding, or subjecting harm on a human fetus. And you can see that he then went on to describe a human embryo as not only a human embryo, but that derived from fertilization, parthenogenesis, cloning, or any of the other methods that I showed you on the previous slide. So that really kept these type of embryonic stem cells out of the federal funding arena as well. And then there was a big discovery by a group in Japan, Yamanaka, who just looked at all these different factors that you could add to stem cell or to skin cells while they're growing and see if you could turn them back towards an embryonic stem cell. To do a real tour de force, he showed that you could scrape skin cells off of a human or a mouse and add these 4 growth factors during their culture ex vivo, and you could make them into cells that were, for all intents and purposes, identical to embryonic stem cells. So they make all of the different tissues of the blood. And in fact, the first proof of principle was shown in a mouse model of sickle cell disease, where skin cells were collected. These 4 factors were put into these skin cells. They were grown to be genetically identical. iPS cells are called induced pluripotent stem cells, so embryonic stem cell-like cells. Then the mutation for sickle cell disease was corrected, and these were transplanted back into these mice, and these mice were cured. So you could take a skin cell, make it into an embryonic stem cell-like cell, turn it into a bone marrow cell, fix the sickle cell disease, and put it back. So that's where things stand now. And then, along comes a new president, Obama, who basically took back, he revoked the previous president's limitations on federal funding involving human embryonic stem cells, and then opened up again research using federal dollars in the U.S. For embryonic stem cells. And in fact, there's now a clinical trial that's been initiated by this company, Geron, testing this approach in humans. They've just treated their first patient. So they get embryonic stem cells, they grow them to neuronal cells, and they're trying to treat paralysis with these cells. So the first patient has actually been treated. But now we have this Dicky Wicker rider that was on federal funds some years ago that says that no federal funds, and this is still in existence, can be expended by the National Institutes of Health for the creation of a human embryo or embryos for research purposes, or in which a human embryo or embryos are destroyed, discarded, or knowingly subjected to the risk of injury or death. So this has been around all along, and we've sort of worried that this might limit things if somebody really tries to interpret it. And in fact, an investigator who was unhappy with not getting grant support for adult stem cell research sued, and this was upheld recently in August. And an injunction against federally funded embryonic stem cell research was granted. That's been temporarily overridden while this is sorted out. So that's really where things stand at this moment. And in fact, there were study sections going on for individuals who were applying for grants to study embryonic stem cells, and there were funds that had already been dispersed for doing studies with embryonic stem cells that all had to be put to a stop. But they're, at least for the moment, back going, but we're going to still have to wait and see what happens as far as embryonic stem cells are concerned. But these induced pluripotent stem cells, the iPS cells, are not subject to this restriction. So those cells that can be made by simple skin cells. So I'll stop there.
Okay. So now we're going to open the floor for questions.
Okay.
If I understand correctly, what has been done or is involved in taking and then adding 4 components, and this is curing sickle cell disease.
In mice, right.
My question is, currently, or could you calculate maybe for another disease it could be done? For example, somebody gets skin and adds other components and cures another disease, like the method.
So I think there are 2 parts to that question. So to cure another disease, it's certainly possible, and we'll see many examples, and there have been subsequent examples. This was the first proof of principle in sickle cell disease. So taking these skin cells, adding these 4 factors, getting them to go back to a very primitive stem cell, like an embryonic stem cell, has been done in many labs now, so it's validated. We've done it ourselves. Most of the time we don't believe things until we are able to do them ourselves. But anyway, we think it actually does work. And these cells can be differentiated to very different types. For example, to neuronal stem cells, so the stem cells of the brain. And in fact, the Geron Corporation has shown that they can make neuronal cells from these embryonic stem cells, and that's what they've done in this trial, and in fact have transplanted the neuronal cells into individuals with paralysis. Another application would be Parkinson's disease, so the cells that make dopamine in the brain that are missing in Parkinson's can be made from these kinds of cells and presumably transplanted. So how far is clinical application away for all of these other disorders depends on really figuring out how to put the brakes on these cells. So one thing that I didn't show, but it's worth mentioning, is that the way that you prove that these are in fact embryonic stem cell-like cells is to show that they can make tumors if you put them into mice. And they make tumors called teratomas, which are tumors that happen in humans, and they're very unusual, but they're the tumors that you hear about that have like teeth and hair and other kinds of mature tissue. And if a cell can make that in a mouse when you transplant it, then it's that primitive. It's like an embryonic stem cell. The problem is if you put those embryonic stem cells into an animal with the intent of curing a disease, you may cause another. And so we certainly wouldn't want to give a tumor to a patient who's paralyzed. And in fact, that has happened. So there was a clinic in Moscow that was doing this kind of work with fetal brain cells, multiple injections. And there was an individual who, after going there from Israel, when he returned, developed recurrent bad headaches, had an MRI of the brain, and it showed multiple tumors in the brain and in the spinal cord. And when they traced the origin of those tumors, it was from the injected cells from multiple different donors. So figuring out how to put the brakes on these stem cells is going to be the big problem in getting this to the clinic.
Thank you.
So the future is now, Doctor. With all the restrictions that you've had in funding, you've been able to develop alternative routes. So do you need any more support, I mean, in terms of money for research, or what kind of support do you think it would be important to receive in order to further your research?
So I personally don't do work on embryonic stem cells. I use adult stem cells. There's nothing controversial about those. We can get those from the patient and modify them, or from a donor. And in that case, they don't need modification, but we can get bone marrow stem cells without any trouble. But for embryonic stem cell research, now that the restriction has been removed to use federal funds, I don't know that it's not going to come right back, and it may. But those who do embryonic stem cell research maintain that they need to have embryonic stem cell lines to work with because that's the gold standard. We know that these cells can make anything, any tissue of the body. And developing the techniques that might, for example, teach us how to put on the brakes so that once we make them into a brain cell, they can no longer make tumors, is probably best worked out with embryonic stem cells. And then applied to the kind that we can make from skin cells or from others. So I think most investigators who've worked with embryonic stem cells in the past, just because they're this new type of stem cell that they can make, it doesn't mean they don't still need funding to at least have these cells as controls, to have these cells as the gold standard by which they compare all of their other types of cells.
I got it that at this stage, this is done in some few centers throughout the world.
Mm-hmm.
So if this is going to be in this sort kind of the clinical trial space, scaling it up to advance on either field. I guess it's about building networks of researchers in other places to expand that, right? What do you envision is the way of building capacities in other places to have large trials that allow to identify the benefits and the harms of the different
So I think that's already happening. For example, there was a study that was published probably 3 years ago now looking at adult stem cells in type 1 diabetes. And that was done in South America with collaboration with investigators here in the U.S. Richard Burt's an investigator who's been looking for many years at using autologous, that is the patient's own bone marrow stem cells, to treat disorders outside of the blood, like autoimmune diseases like lupus and rheumatoid arthritis and type 1 diabetes. And so I think that infrastructure is already building. That's with adult stem cells. And I think once that infrastructure is built, then logically the next step would be to begin to test iPS cells, these induced pluripotent stem cells, or even embryonic stem cells if they reach the clinic in a broader sense than what's available now. This infrastructure will be valuable in moving those trials forward.
Are European scientists allowed to use stem cells in their research? Do you have any limitations like in the U.S.?
It depends on the country. I'm not sure about all of the countries, but there are many that have similar limitations in the use of embryonic stem cells. There was, at one point, a stem cell center opened up in Singapore where there were no restrictions. So there were a lot of U.S. investigators that I know that actually left the U.S. in order to be able to receive those funds to continue their research.
You're having a brain drain in the opposite direction.
Yeah, that's right.
Yeah.
That's right.
I have another question.
Okay.
Okay. You mentioned a couple times infrastructure, responding to his question. And I was going to ask you, can you tell us a little bit about the infrastructure that you used for your research, the team that involved, what was the team, and how long it took? And you can give an idea of how much money was total like.
So for the one study that I showed you, we started off more than a decade ago with an interest in whether we could do transplants without ablating the bone marrow. That means, high doses of chemotherapy and radiation that completely knocks out the bone marrow stem cells that are there, and then you give the new ones from the donor, and they grow up. So we had this idea that we didn't have to do that to make it work. And we actually started a clinical trial in humans with a different disease, a rapidly fatal disease, kidney cancer that had metastasized. That's about a 6-month survival usually. And we figured out that you could do that, and the grafts do take, and some patients had their tumors go away. So that was a big home run. But it didn't look like it was good enough for sickle cell disease. So we had to go back to the mouse, try new transplant studies in mouse models to tweak the way that we get the cells in enough that we felt it was safe enough in patients who weren't at immediate risk for dying, like kidney cancer, but were, risk for dying early, say 40 years of age. And so that took probably 5 or 6 years to get all that worked out in the lab. And then we had different ways of collecting the stem cells from the patients, so that was a 2-year study in volunteers. And then we opened the clinical trial for patients with sickle cell disease, and that took an additional 3 years. And that funding was probably, I don't even know. It's at least at least our own budget for doing that is probably at least $1 million a year over 10 years.
The monkeys, you can mention them.
Yeah, so we didn't do monkey transplant studies for the sickle cell part, but we also have so we usually go from the idea to the bench to small animals, mice, to larger animals, monkeys, then to humans, and then usually right back to the bench, because we always find that there's something that we can do a little better. And that's about a 10-year cycle.
So currently, what conditions are being mostly researched? And if one has one of those conditions and the situation where you think you would like to participate in a trial or a study, how does one engage with the trial? So how does one learn what's going on and whether you can participate?
So I think that the two biggest interests right now for stem cells are diabetes and neurologic disease, so spinal cord injury. I think these are going to be the most difficult too. So it's perhaps unfortunate that that's the greatest interest, because I think those have the highest likelihood of failing. And when something has had as much hype as stem cells, if you fail the first time you go to the clinic, then it's really bad for the field. So, but those are the two most likely to be in clinical trials.
Why do you think they have the highest likelihood of failing?
Because they're much more difficult. So to think that you could grow some neuronal cells and squirt them into somebody's back after a spinal cord injury, it's a long stretch between squirting them in and them actually, becoming incorporated into the spinal cord, repairing whatever the damage was in the spinal cord. Whereas with something like Parkinson's disease, we know that there's just a certain cell that's missing in the brain that makes dopamine. Okay? And if we give exogenous dopamine, that helps. But if we can give it in the brain, it restores them to normal. There are animal models that show that if you put a few cells in that can make dopamine, they're completely fixed. And in fact, you can do it on one side. And there are monkey models of this where they have Parkinson's on both sides. They treat one side, not the other. And you can see the animal. You put the food in two different boxes, one with the right hand, one in the left, and they'll reach with the right hand over to the left box if you fix the right side and the food is on the left. So, it's really clear that that's something you can measure, you can demonstrate that it works well. But for whatever reason, spinal cord injury is one where there is a big push from the community because it's so devastating. For example, to have a child who's had a car accident, been involved in a car accident, no longer walk. If you could make them walk again, that'd be terrific. So there's a big push from the community, I think, that drives that that's the first place that this went. And there's a company that has a patent on embryonic stem cells, Geron, that if they get into clinical trials, they can keep the patent alive. So there are other issues, I think, that push it that way. But and diabetes should be easy, but it's just been very difficult to grow the cells that are missing in type 1 diabetes from embryonic stem cells. So I think that's also going to be hard. But those are the two big ones. Now, how do you access clinical trials? There's a site, clinicaltrials.gov, where you can just type in the disease and you just have a big list of all the active and recruiting clinical trials. with all the contact information. And so that's a terrific spot for you.
Very short question. For the diabetes and for the spinal cord injuries, only embryonic cells, or can you also use adult cells or a combination of?
So there are people that are working on, there's three ways that people are working on it. One is embryonic stem cells. So in type 1 diabetes, the beta cells that make insulin have been attacked by the immune system and are gone. So you want to grow new ones. So if you could grow them from embryonic stem cells, that would be great. Others have tried to grow them from adult stem cells. We've also tried that ourselves and found that to be a very difficult proposition. So far, we've had no luck in making adult stem cells grow to those beta cells. The third way is to try and get some other cell in the body to change, to take over the role of a beta cell in the pancreas. And that may end up working, actually. So liver cells and pancreas cells split off sort of late in embryogenesis. And so they have a lot of the same properties. And so some people believe that it might be easy to turn these liver cells back into the cells that make insulin. And so there have been people working on ways to switch at least a portion of the liver cells to be sensitive to sugar and to make insulin in response to it, like a normal beta cell does. So those are the three ways.
And that is more gene therapy rather than stem cell.
Right.
Yes.
Right.
I have been following this through the Illuminate, and then I said myself.
A little louder.
Sorry.
The microphone is over here. That's really great. Thanks.
I'm Gamo Vidal from the Medicines and Technology Unit, here with my colleague Jose Luis Castro. I think that you mentioned like several issues which are really really interesting, especially like when you refer like to the policy change in terms of public funding and the fact that there are private companies working on it. And you mentioned like the taboo work of patent. I work on IP issues. On IP issues and public health.
Yeah.
And I think it's really interesting to see this, how it would, like in the future, affect access to whatever product is achieved, whether it is in the private sector or public funded research. And I think that you were really, you nailed it, no? When you said there's a push by the community, in the sense that there is a demand, no? Especially non-communicable diseases, cardiovascular, degenerative. And what do you think we could do now when we are at the earlier stages of research to secure access to these products, which will not be cheap, that's for sure?
Well, I think you get my opinion now, and that is that this type of research should be federally funded so that it's open to all. Because if it's developed in the private sector, it will remain in the private sector, and it will be a money-making venture from start to finish. And that will make it difficult to access, especially in places where there's not a lot of money. So to me, that's a big problem. And that's a big problem with the way this research has been pushed out of the public and into the private sector. So if federal funds could be brought to bear, it would also increase the amount of funding available so that progress could be made more quickly. But the reason why I sort of intertwine the story between policy and science is because I don't think there's ever been a time when policy affected more the science than in this case. Because you could see, okay, this isn't covered, so let's try, fusing these cells. And this isn't covered, so let's try taking the nucleus from here and putting it there. And then the next directive says, okay, you can do everything but those things. But I mean, actually, something good did come of that, and that is that these iPS cells seem to be the real thing. And at the same time, embryonic stem cell research was funded for the majority of that period. So I think what Bush said was, okay, so if these cells are great, prove it with the ones that you already have. I mean, that's not necessarily a bad position. If you've got 60 embryonic stem cells line to study, that should be enough for you to show me it works. It just turned out that wasn't the case. There were really only 4 or 5 that were reliably available.
Following on that question, I understand from the presentation that the ones that are working now are the more complex ones. What you were saying about medullary injury and diabetes. But on other diseases like blood disorders, there's already some advanced and actually some treatments that are being offered in different places. So I'm just wondering, what is the path that follows for example, in sickle cell disease? That is something that you've worked a lot. When you go to I work in the Pacific coast of Colombia, and 8% of the population basically had the trait, and 1% of the people roughly were affected by the disease itself. How long do you see that happening? What's going to take to get treatment accessible to those people if the treatment is really working the way that this suggests is going to work?
So it's going to take at least a decade more of work to get this even large animal for making embryonic stem cells, like the iPS cells, into blood type cells, fixing the mutation, and then putting it back. Because there's been a huge problem in growing blood cells from iPS cells. So that's been an area of active research. So human cells seem very resistant to that transformation from iPS to blood. And that hasn't been sorted out yet. So that'll have to be sorted out. Then there'll have to be safety issues sorted out. So these are viruses that are used to put these genes in. When these viruses integrate into the chromosome, they can turn on tumor genes. So they have to be screened to make sure they're not next to genes that might cause tumors. So there's a lot of safety experiments that have to be done before this could be moved. So I think what I showed you in the mouse is at least a decade away from even large animals at this point. So it's the sort of thing that's going to take a lot more time to move to the clinic. But we have adult stem cells for diseases like sickle cell disease that we now know work. So if we can use a brother or sister as a donor, that could be exported now to these areas.
And are the effects inherited? Like once you get your, let's say, if I have the trait or the disease and I get treated, but my kids, will they have the disease or will they, let's say, preemptively cured?
So if you have the disease, that means you got the gene from mother and father.
Yes.
And so now you have the disease. It's autosomal recessive, so you have to get both genes. So now you have the disease. You go get a bone marrow transplant, so your bone marrow is now fixed. So your bone marrow no longer has the gene. But the rest of your body still has the gene. So if you have a child, you'll pass one copy to that child. So if the mother of that child also has sickle cell disease or trait, then there's a chance that the child will have it. If the mother has disease, the child will. If the mother has trait, it's 50-50. It doesn't fix. And none of these types of therapies would fix anything but the tissue affected. So you would still pass the gene on.
Okay.
Question, doctor. Some of the situation stays the same in terms of support. I mean, like, are you in a position to tell what can we expect in 30 or 50 years?
No, I think people will move more and more towards supporting this kind of research. The same thing happened when DNA was discovered. You know, you shouldn't touch DNA, you shouldn't isolate DNA, and there was a real pushback. And it's fun to go back and read some of those articles from the time when DNA was just discovered. So we've moved past that. I think we'll move past this as well. So in 30 or 40 years, I don't think we'll be having these conversations, but I think the cells that we have now are sufficiently free of ethical concerns that we can move ahead with the IPS.
And what prevents the development being done elsewhere?
I think at the moment these are very difficult to produce cells that require a lot of reagents that are expensive, and laboratories that are fairly high tech, so that those types of laboratories just don't exist in many places, even in the States. There's just a handful of places that can actually make these cells. So that, when these iPS cells, for example, were first described, many people didn't believe it because they tried over and over and over and couldn't make it work. So finally they got enough expertise and people working on it that were able to make it work. But I think that's the main reason. It takes a lot of money, and it takes a very highly technical lab to pull it off.
There are those plants that for a tea they keep the umbilical cord to be used in case of a disease. So what kinds of diseases can they be used if the child gets?
So the disease that I study, sickle cell disease, it can be used in that way. In fact, we have a protocol at the NIH where if you're a couple and you're at risk for having a child with sickle cell disease, so you have trait, your spouse or significant other has trait, or both have disease, or any combination of that, we will consent them so that at the time of delivery, we can send somebody out to collect their cord blood, freeze it viably, so that if they ever need a transplant, we have those cells. Now, cord blood can repopulate the bone marrow. So you can take cord blood and do a bone marrow transplant and replace the blood. And in fact, we had a child who was born with sickle cell disease. We saved his cord. A subsequent child in that same family was born without, and the second child had a transplant with the first. There's also the possibility that those cord blood cells can be used and corrected. So the other thing that we work on in the lab is actually to take bone marrow stem cells from people with a genetic disease like sickle cell disease and correct that mutation. And the way that we do that is to use a virus to transfer the correct gene back to those stem cells. And that will probably, the protocol is now written and through all of the hoops. In fact, we just had a long conference call yesterday about starting within the year doing that technique in adults with sickle cell disease and thalassemia, which is a related disease. And the gene that we use, the virus that we use to transfer that gene is actually HIV. So it turns out that it can be quite useful in some regard. So we take it and gut it of all of the infectious components and then put the gene in that is missing. And in this case, it's the gene for hemoglobin in sickle cell disease. It's misspelled. We put the correctly spelled gene in place of those infectious elements, and then the outer part of the virus will in fact transfer it to bone marrow stem cells if we put them together in culture for a day or two.
I have one question about cost. So we have, obviously it takes a huge team and a lot of money to find these results, to actually get to the results we want, which is to cure sickle cell in this case. In the case of, say, diabetes, where obviously for somebody who has type 1 diabetes, if the stem cell research or the gene therapy were to work for them, for type 1 diabetes, yes, you have it from the moment you're born, so it is economic to deliver that?
No.
Okay. So would it be, for them, it would save them money to just, I guess, do a one-time therapy deal. But for somebody who has type 2 diabetes, I think it would depend on how much their treatment or the insulin will cost them in the long term compared to how much this therapy will cost them. Can you tell me anything about what, in the future, what you foresee to be the cost of these for a patient compared to the actual treatment?
Yeah, so
actually,
it depends on how it works out. So for sickle cell disease, these kinds of calculations have been done already. So the lifelong cost of sickle cell disease is pretty extraordinary because they're in and out of the hospital sometimes every month. And it's hundreds of thousands of dollars a year. There are other kinds of diseases where there's enzyme replacement therapy that costs $100,000 a year. So
when you start to compare against those sorts of costs, techniques like a transplant don't seem that expensive. A bone marrow transplant at a regular hospital is probably $200,000. These kinds of therapies, where especially if it's, let's say you have Parkinson's and you want to make some cell fix that, and so you go somewhere and they scrape some stem cells off and they grow those, and then they make them into iPS cells, and then they grow those into neuronal cells, and then they implant those into your brain. That's likely to be very expensive because that's individualized treatment. And I think that may be required at first because if you were to take my embryonic stem cells given to you, your body would reject them. So it would have to be individualized. And I think the cost of that would probably be so. There are efforts about now to try and develop cells that might, like a bank of cells that have certain HLA types, that's sort of the immune fingerprint, so that we have most people covered, and there are 1,000 or so embryonic stem cells developed. There may be enough that we could cover most people, and that would reduce the cost significantly. But that would still leave out significant fraction of patients who might otherwise benefit, but then they could have their own. I think it's going to be, at least for the short term, expensive proposition for just about anything.
Okay, no, I had a question. Not really about stem cells, but you mentioned something about how right now there's a lot of debate for stem cells. I think usually for DNA, that DNA is not. So what happened with DNA now? Because there is no debate. Has it really given fruit for everything that it was said it was going to be done? Because some might have, and I don't think we know a lot about this, but some might have that, yeah, after all, there was still a long way to go to actually DNA to bear fruit.
So when I was talking about the controversy with DNA, I was talking about when it was first discovered, and the code of life, and so everybody was like, "Oh no, we can't touch that." That was a controversy I was talking about many years ago. But now you hear a lot about how all of these techniques that have been developed to be able to sequence DNA, and now we know the human genome sequence. And if we just know the human genome sequence, we're going to be able to fix all of these diseases. That's right. There hasn't been much delivery in that regard. So I think there's still a lot more work to be done in having this genome era bear fruit. But there have been many things developed, like our ability to transfer genes. If we didn't know the genes, if we didn't know the genetic defect, we wouldn't know what to put into there and fix. So this really has, there have been some fruit. Certainly not as much as you would expect by the popular crowd.
Does crossbreeding and mixture of races have any effect on all this? Like if you have a...
Yes. So in the case of sickle cell disease, it does. So, for example, if you need a bone marrow transplant in America for leukemia and you're Caucasian and you don't have a brother or sister that matches, you can find a donor in the unrelated donor registry. So this is where people go and donate a sample of blood. They have their HLA type catalog, and that's, again, the fingerprint of your immune system that allows transplant to happen. You can go find a donor. I mean, I could find a donor easily. There would probably be hundreds in the donor registry. But for African Americans, it's nearly impossible. It's harder to find a donor in the unrelated donor registry than it is in the family. It's always the other way around for everyone else. So this mixing makes for very distinct HLA types that are hard to match. So at least for transplant, that makes it difficult. It's good for the immune system, actually. The immune system usually functions better the more disparate the native.
We can maybe do one or two more questions, and then the time will be up.
How about that, what you were mentioning about antigenicity of stem cells? same problems as in adult cells. I mean, there is no chance or more flexibility for managing these anti-initiative stem cells.
So,
or kind of redirecting or something.
So there's been a lot of talk that stem cells will be easier to transplant because they don't express a lot of these antigens that cause rejection. But the problem with that thinking is that no one would transplant the stem cells in these diseases. In fact, as I said before, the stem cells are the ones that make the tumors. So the strategy for everyone is that they grow these stem cells to the mature type tissue and then transplant that mature type tissue, and those mature cells will express the antigens that will cause rejection. So unless you're taking the cells from the individual, making them, and then giving them back, then you have the additional barrier of the immune system rejecting them. So that will be something else to overcome. But there's been work in that regard as well, and some of the work that we did to make transplants work for sickle cell disease can be used to make patients tolerant to tissues that have been derived from others.
Anybody one last question or? Okay. Well, thank you so much for coming to talk to us today. It was very informational, and hope to have you here again in person.
Thank you.
Good. I'm serious.